So we have to move to Dr. Kumar from India, National Institute of Immunology. His talk will be about inhibitory and the disintegration effect of Ocimum sanctum extract on α-synuclein aggregation. Now you can share.
Hi everyone. Let me start sharing.
We can hear it. I can hear you, and you can share. Yes, we can see. Go ahead.
So I was checking it's okay or not. Good day everyone. Myself Vijay Kumar. I am presenting our research inhibitory and disaggregation effect of Ocimum sanctum extract on α-synuclein aggregation. Alpha-synuclein is mainly responsible aggregation mainly responsible for Parkinson disease. So let's start with Parkinson disease introduction. It is the second largest progressive disease neurodegenerative disorder of CNS after Alzheimer's disease. It was first described by Dr. James Parkinson in 1817 by shaking palsy due to the uncontrollable movement of limbs, and it affects the NSPC of the brain, which mainly control the movement part of our body, and it affects mainly 6 million people in people in 2016 as reported by GBT, and notable cases are the actor Michael J. Fox, boxer Muhammad Ali, and athletes Davis Finney. So what are the causes of Parkinson disease? There are not specific mentioned what are the responsible. There are some 15% of cases which are in the hereditary region and the genetics region for the aggregation for the Parkinson disease, but there are some studies which show that pesticides, air pollutions, and the mercury, which are the mainly found in trapped in our food cycles, can also cause the Parkinson disease. There are two types of symptom of the Parkinson disease. One is the motor symptom, which can include tremor, bradykinesia, akinesia, and gait. These are the mainly rigid body movement and uncontrollable body movement. But in non-motor symptom include depressions, hallucinations, and sensory and sleep disorders. So what is the prevalence of Parkinson disease in around the world? And so there is a study published in Lancet Neurology in 2018, which conclude from 1996 to 2060 that most of the cases are present in higher case number of cases are in the Canada and US as compared to the South Africa. There are very less number, as you can see here. And there are also gender with increasing the age, there are increasing the Parkinson disease patient number, which can also seen in previous this data that the life expectancy in Western countries high and similarly in case of Parkinson disease is high in case of male as compared to the females. The type of Parkinson disease on the basis of how they occur, there are two types, familial and sporadic. In familial, there are mainly transfer from one generation to another generation caused by mutations, gene duplication and triplication. In the sporadic type, they are not specified why they happen, and there are no specific reason. There are some reason are the aging, neurotoxin, and environmental factor which cause the Parkinson disease. What are the molecular mechanism in Parkinson disease? There is alpha-synuclein is firstly bind with some environmental toxin or caused by aging or oxidative stress. It misfolded up its normal physiological state and form a oligomer or small aggregate which act as a nuclei and further form the alpha-synuclein fibrils. What is the alpha-synuclein? Alpha-synuclein is the first gene to be known with Parkinson disease. Is found in the Levy's body, which are the cause in the SNPC part of brain found in the postmortem of Parkinson patients. So it have alpha-synuclein have three region: N region, N terminal, C terminal, and neck domain. The C terminal is supposed to be it's a move like pendulum in structures. I will show later, and it bind with a neck domain and stop its aggregation. And in the neck domain, there are 12 amino acid residue which cause the mainly responsible for the aggregations. And the function of alpha-synuclein, there are not specified what are the not specifically mentioned because it is unstructured protein. So but its presence in the presynaptical vessels, it's considered to be involved in synaptic activity, synaptic plasticity, neurotransmitter formation, vesicle trafficking, and recently studies saw it shows chaperone-like activity with B snare and T snare, and fibrillization of alpha-synuclein stop all of these processes, so it causes the cell death. The how aggregation of alpha-synuclein occurs, so we can stop it. So it can divide it into two pathway, O pathway, on pathway. In when monomeric alpha-synuclein start unfolded and interact with another monomer and form oligomer. These oligomer are two type: amorphous type, which are the O pathway pyrgryl, they are not toxic in nature, and on pathway they are form oligomer, which are act as a nuclei, which further form protofibril and fibril, and which goes on, and they are the toxic in nature. So what are the strategies to stop the alpha-synuclein aggregation? First strategy is to maintain alpha-synuclein its native form, and further if it's formed oligomer, direct it toward the O-fibril pathway, so they form the non-toxic pathway, non-toxic oligomers. And another is, if there is pre-formed fibril, we have to stop, we have to disassemble them and disaggregate them into the non-toxic one. What is the rationale behind this? There are currently no preventive therapy available for PD. And the only available drugs are carbidopa and levodopa, which are the analog of dopamines. And there are unmet need of developing novel agent that can modulate, inhibit amyloidogenesis pathway. There are studies show that phenolics compound can prevent the alpha-synuclein oligomerization and fibrillization and reverse this toxicity. But why is Ocimum sanctum? We start with many plants, but Ocimum sanctum show them the higher activity, and there are various studies which show that Ocimum sanctum have effect on the neurodegenerative disease, and it protect the neural cells from the various disease like antioxidant and the injuries. So we start with the purification of alpha-synuclein. So first we start with by acidic precipitation purified, and then we just done gel filtration chromatography. Here it shows the monomer peak. We took this peak in our samples and run in on SDS page. It show higher than the 15 kDa, which is norm above than its size, because its size is 14.4 kDa. So it's above due to the acidic high acidic residue in sequence. So we check their protein sequence, check its molecular weight. It's come about similar with MALDI. It's come similar to theoretical value, and we've done a mutations, one mutation, and it's also come nearly equivalent to the theoretical calculated part. But then we start the purification of Ocimum sanctum extract. So we take its leaf, wash, dried, and crushed in mortar and pestle, and then we crushed the crushed part mixed with methanol in 1:10 ratio and retort for 24 hours at 37 degrees Celsius in closed flask, and then we methanolic extract solution were filtered and allowed to evaporate, and the remaining fraction were dissolved in DMSO for experiment. So first we check that if there are no effect of chlorophyll or a DMSO on the extract. So we did a TST fluorescence assay. In TST fluorescence assay, the TST thioflavin T bind with the fibril part of protein and show higher fluorescence. So we check that we add alpha-synuclein alone plus alpha-synuclein plus chlorophyll plus alpha-synuclein plus DMSO, and we show that there is no significant change in the its aggregation process, and so the DMSO and chlorophyll does not affect the alpha-synuclein aggregation effect. To check the inhibitory effect of alpha-synuclein aggregation, we took THT fluorescence for we did aggregation for 7 days, and here you can see that without without OS extract, the alpha-synuclein form a 2-day lag phase, then start log phase for 2 days, and then further go for stationary phase. But in presence of OS extract, it only remain in the lag phase. So it's inhibited. To check that it's how the fibril or oligomer have the higher hydrophobicity. So we check the ANS fluorescence. ANS dye bind with the hydrophobic patch. So with monomeric alpha-synuclein, there is very less binding. But when alpha-synuclein we did for 7-day aggregation, it bind very high. And when a presence of OS extract for 7 days, we check it's around 80% decrease in hydrophobicity compared to the alpha-synuclein alone. The Congo red is another dye which bind with the fibril and show higher absorbance toward around 420 to 440 nanometer. But when in presence of alpha OS extract, it's so similar to the monomeric toward the monomeric seal. So to check how they bind OS extract, we took the alpha-synuclein 35 micrometer and we did tryptophan tyrosine quenching assay. So for that we took 35 micromolar alpha-synuclein and add increasing concentration of OS extract. Here you can see the blue one is here is the protein only, with increasing concentration from 1 to 1:35 micromolar to 420 micromolar. The tyrosine fluorescence is quenching, which suggests that it binds mainly with the tyrosine part, tyrosine or near tyrosine part. To check how, I always suggest if it make them keep them monomer, it's a good strategy. To check the OS extract effect on solubility of alpha-synuclein, we run the 7-day experiments on the alpha OS extract on into 1 to 4 extract alpha-synuclein to OS ratio 1 to 2 and 1 to 1. In this gels you can see it is a monomer, and in the presence of 1 to 1 ratio there are a high amount of alpha-synuclein monomer we can see near this, but in presence of 7-day aggregates there are no monomer band C, so they all stuck in the stacking gel itself. So further we check the solubility. To solubility we check the turbidity of our solution mixture. So here we can see in the presence of OS extract the alpha-synuclein is very much soluble and fibril are not soluble, so they show higher absorbance at 360, but in presence of OS extract they show very less OD absorbance. And similar with the light scattering experiment, in the presence of OS extract it show around 50% decrease in light scattering at 350 nanometer. To check that how it affects the size of, because size is the one of the main thing which can we decide it's fibril or oligomer. So it's attenuating. To check, first we did the TEM assay, transmission electron microscopy. So for that we did after we take alpha-synuclein for 7 days and alpha-synuclein with OS extract 7 days fibril assays and put them check their assay. So here you can see there are the long rigid fibril and shape, but in this you can there are very less fibrils and they are mainly amorphous in shape. They are not in fibril shape. To check further its size, we took the DLS data, dynamic light scattering assay, and in that we can see in the presence of OS extract the size of oligomers after 7 days is around 50 to 90 nanometer, but in alpha-synuclein alone it around 800 to 900 nanometer. To check if there are preformed, as I suggested earlier, our strategy, if there are preformed fibrils, so can it affect them and degrade them? So we took the preformed alpha-synuclein fibrils and to control we took one without the OS extract and one with extract. So with extract it decrease is OS intensity, which so that it's disaggregate and disassemble the preformed alpha-synuclein fibril. It's a 7-day alpha-synuclein fibril, and after 5 days with OS extract, they show amorphous type aggregate oligomer to check how they affect the toxicity of cell viability or toxicity of this oligomer and fibril. To control there is cell viability and to control, and alpha-synuclein alone itself can cause, because in its very aggregate from very aggregate very early in stage, so it shows very less significant compared to the buffer, and similar result we saw with fibril or oligomer formed after 7 days, and the disaggregate fibril after 5 days compared to the alpha-synuclein. In case of this, it around 90% cell viability in OS extract. To check what are the molecular mechanism or what are the molecules present in it, we did the MS analysis, and there are MS analysis. So for MS analysis, we identified three, various molecules which are present in it by checking the literature, various literature available for that. So we found these are mainly. So then we further checked which are the molecule can be responsible for the aggregation inhibition. So for that we took six compound: rosmarinic acid, crustine, epigenin, AGCT, methyloeugenol, and ascletin. So ascletin does not showing to be its kind of negative control, but in case of after leaving rosmarinic acid which attached to the C terminal part of, all the other part bind with the N terminal and neck region, which are the main region responsible for the aggregation help aggregation. These are the preferentially bind, and as we can see, rosmarinic acid which bind with the C terminal bind with the highest affinity, and the methyloeugenol bind with very lower affinity. So in some research, it show that it affect the fibrillization process, but for eugenol, but not for methyloeugenol. And for check the their familial, there are the sporadic. First we did the on the sporadic to check if it affect OS extract affect on the familial type aggregation or Parkinson disease process. To check, we did normalize TSTSA. So we did with it alpha-synuclein with mutant. So mutant does not have that much lag phase, because in normal wild type alpha-synuclein it have 2 days around lag phase, but in case of mutant in familial type, it's start in lag phase in 1 day, which is the reason it in familial type it start from the 40 years, but in normal sporadic it start from the 50 year and above age. So after 5 days with aggregation with mutant type, it's so that it can aggregate 25%. Around 75% of the aggregations, which is less than the wild type, which are 20, which is 80%, but it's significant. Let's summarize this. In summarize, we use OS extract effectively inhibit modulate the aggregation of wild type as well as alpha mutant. OS extract maintains solubility of protein for longer duration, and compound presenting OS extract interact with N terminal and neck domain of protein. OS extract and associated molecule may become potential therapeutic intervention against Parkinson disease and other aggregation related disorder. And these are the this is the first paper which I present the data. Similar data we showed within this paper with ellagic acid compound. And thank you DBT and NII for financial help, and Beyond a Science Initiative for presenting my data. Thank you.
Thank you.
Is there any questions? I'm happy to take it.
Yes. Attendees say interesting talk with the promising data for the OS extract. There are several questions. What small animal model of Parkinson's will you interrogate for the benefit of the OS extract?
Actually, our lab does not have expertise in the animal modeling or a kind of, but we did on the cellular level, and now we moved with some compound from we extracted from here, and we purchased from market, and we moved on the another similar protein which from the aggregate like SOD, which cause amyotrophic lateral sclerosis, or Alzheimer disease, which caused by the A beta proteins. So we moving on them.
Okay. How could you show that the extract is effective? How could you prove it?
There are previously studies which on the Drosophila models, which show that it affect very much their movement and reduce very much symptom. It's mainly preventive, not technically. I can't say for the sure for disaggregation part or as a drug, but it's preventive. So we can take it on or diet on daily basis. And it's herb, and it's Ocimum sanctum is also a herb disease. It have various benefits like anti-cancerous, antiviral, antimicrobial its effects. So we can take it on daily basis. In our tea, there are many teas, green teas, which provide OS extract or OS leaves in it.
Okay.
Maybe just a quick question. Vijay, really great talk, and actually really nice to see somebody presenting from NII. I'm not sure if you know, but it's one of the first places that BSI was active. So great to meet you, and thank you for the great talk.
I know that some of my senior told me that it start with in India from National Institute of Immunology.
Yeah, so I'm just curious. It's really interesting your work. I'm curious. I'm sure there are more such extracts out there. What do you think is a good way to do a screen and a big survey of what could be effective?
We did actually with 10 plants, 10 medicinal plants which are recommended in Ayurveda, which we used on our local element techniques. So, but we found that Ocimum sanctum, which is only also known as holy basil, if someone not its common name. So it's it affect more, it reduce more aggregation process compared to the other process. We also go with aloe vera and Azadirachta indica, which is neem, I think. So they also reduce, but not as much as this. They reduce around 60%, but this reduce as 80%. So we go for this.
It's very cool. Thank you so much.
Okay.
Thank you.
Professor Eleanor is asking you to pass our greeting from PSI to NII. Okay.
Thank you.
Thank you.